regulatory T cells, they are a population of lymphocytes. So more exactly, our immune system, it's protect ourselves from invading microbes, but it doesn't react with our own tissue. If it reacts and then destroy our own tissue, it causes autoimmune diseases. So how it is called immune tolerance or self-tolerance, and how it is established and maintained. That has been a key question in modern immunology. There have been proposed many various mechanisms, and one of the mechanisms that is the uh, immune suppression by regulatory T cells. So they are present in our body, and if the number or function of regulatory T cells reduced, then the immune system turned on to our own tissue, causing autoimmune diseases. So regulatory T cells they are key lymphocyte for maintaining immune tolerance. Well, a majority of them they are produced by the thymus. So the thymus produces T cells. So T cells mediate various immune responses. But also the thymus produces such the immunosuppressive lymphocyte regulatory T cells. So both the those T lymphocyte mediating disease and then also T cells suppressing such the immune responses, the magnitude, and then it's just proper, not too much or too little. So that is kind of, well, main function of regulatory T cells. It was not bad idea that lymphocytes, T lymphocytes, and the mediating disease, and also another population of T lymphocyte which control the immune responses, not to be, not to be excessive or properly directed to. So it wasn't a bad idea, but the people, immunologists, they failed in defining that population. So look, this is that population. So everybody can look at this with this marker or something. And so no such success in research. And then also, I think it's more that is more important. That is, if anomaly of suppressor T cells, it is responsible for, say, autoimmune disease or allergy, some immunological diseases in humans. And then people can go on in studying this population. But it didn't happen. And especially when the molecular biology technology developed, and then scrutinized the genetic basis of that suppressor population. And then all that kind of attempt failed. So that was the main reason that people now became very suspicious about the presence of such cells, existence of such cells. And then immunologists walked away. But I was interested in not suppressor T cells, but in autoimmune disease. So whatever the mechanism is, if we can explain the autoimmune disease, the mechanism or cause, that is what I wanted to learn, or wanted to know. So we analyzed the autoimmune disease models and then came up with, well, the population of lymphocytes which are specialized for immune suppression. If that lymphocyte has some anomaly in function or number, then you'll see autoimmune disease. And if you put back such lymphocyte, and then you can prevent or suppress disease development. So that was my start working on this population of cells. We were convinced that this population is important, but other immunologists still they were suspicious about that kind of cells. Well, suppressive cells. That's a kind of dirty science. So, but we have been well, but we could convince ourselves by looking at, well, look, the autoimmune disease really happens very similar to the human counterpart by manipulating the regulatory T cell and removing them. And then we see autoimmune diseases and put it back, and then autoimmune disease can be prevented. So always we can go back to that finding. If we failed in the experiments, but that finding very solid and then very reliable. So that was the perhaps good reason that we have been able to continue anyway working on this population of cells. Yes, we can. I mean, so let me tell you the story. So we show that Tregs are present in experimental animals, but we didn't know, we couldn't show that, well, this mechanism is really responsible for a real human disease. And then in the year 2001, the two co-Nobel laureate last year, Fred Ramsdell and Mary Blanco, these two people, they cloned a gene called FOXP3. And then that FOXP3 gene is responsible for human disease called IPEX syndrome. So in IPEX syndrome, autoimmune diseases really rampantly happen. So more than 80% of the patients develop type 1 diabetes and other autoimmune diseases such as thyroiditis and others. And then 100% inflammatory bowel disease and 100% allergy, including food allergy. So, and then it turned out that the FOXP3 is specifically expressed by Treg. So animal experiments and then human finding altogether, and then now we can say that the Treg, they can be responsible for human diseases, including autoimmune disease, allergy, or inflammatory bowel disease, if Treg number or function become altered or diminished. And at the same time, it means that this population can be a key target for prevention or treatment of these immunological diseases. So many clinical immunologists started to look at the real human diseases, not such rare IPEX syndrome, but the autoimmune diseases which afflict, say, 10% of the population, common autoimmune diseases, including type 1 diabetes and others. So whether Treg number or dysfunction really present in these diseases, or by expanding Treg, can we prevent or treat these diseases? So that is still a trend in Treg research, and many attempts have been made for that, in that direction. So still we are in the middle of developing a Treg-based therapy, immunological diseases. Our aim is disease-specific immune suppression. That is our ultimate goal. So as you may know, currently good immunosuppressive drugs are available. For example, steroid, yes, or tacrolimus or cyclosporin A. All are very good, very effective immunosuppressive drugs. But they are antigen non-specific. So all the immune response can be dampened. So this means that, well, it's good for treatment of autoimmune diseases, but at the same time, make the patients more sensitive or vulnerable to infections because such antimicrobial immune responses can be suppressed as well. So the question is how we can suppress particular bad immune responses such as autoimmune disease, sorry, or allergy or inflammatory bowel disease, but not the antimicrobial immune responses. And the regulatory T cell, Treg, they are T cells, so they can recognize the antigen. So if antigen-specific regulatory T cells, the Tregs, if they can be expanded in vivo, that is the best way, one of the ideal ways of antigen-specific treatment of autoimmune disease. So now the many clinicians or clinical immunologists, they try to expand naturally present Treg present in our body, how we can expand them. And then, for example, ex vivo means in the test tube, and then transfer back to the patients. So that is one way, and then lots of attempts have been made. And then also we are a bit more ambitious. So if the disease is mediated by T cells, if we can convert such bad T cells to Treg, that is the best way, one of the ideal ways of immune suppression. So why we think that this is ideal? Well, because the T cells mediate disease, but we don't know in many cases which target antigens, which self-antigens are targeted. Sometimes it's so many. For example, type 1 diabetes, more than 4. And then also it may be different among individual patients. So SLE, more than 10 target self-antigens. So, and then inflammatory bowel disease, still we don't know the real target antigen, either self-antigen or microbial antigen, we don't know. But T cells are involved. And then the question is, whatever it is, whatever antigen the T cell recognize, but if they are mediating disease, and then if we can convert them into Treg, that is then if you infuse back to the patient, eventually you can establish the antigen-specific suppression because T cells, they have an antigen specificity. And then with that specificity, now Treg, with that specificity, can go to the inflammation site and suppress that particular immune response.
So you are kind of turning the bad guys to good guys.
Yes, exactly. Well, it's still we are in the middle of developing such technologies. But now we are at the stage where we can apply for real human diseases. So, of course, it is great honor for me. But there are so many important researches in the world, and excellent immunologists, so many. And then, in a sense, I'm a kind of lucky person to be chosen for the award. And at the same time, I see that if I can do something as a Nobel laureate, well, so if I can say something or propose something to, for example, to our government in Japan, and then if we can help basic sciences in Japan, perhaps people may expect me to do so. So after I was awarded last December, yes, and this year several times I visited the government and the Congress, and then appealed to the government and then the Congress people that, well, the support for basic sciences should be increased. Well, we are under kind of inflation, and then so the value of the research fund, it's becoming less and less. So now this year, a bit now increased. So I think it worked. So what is important is he or she has his or her own interest, strong interest in something, in science, and then continue. So and then he or she may find that her idea or his idea is good or bad or a bit modified, but then just for a while, what is important is to continue working on that. And then also another thing is to be optimistic in science. That is important. That is important. As far as I can tell, the good scientists, they are all optimistic. For example, in my case, so I started working on a mouse model, and as I told you that, well, how it can be extended to human diseases, and then very fortunately, the Mary Blankoff and Fred Ramsdale, these two people, they found the gene FOXP3. So it was unexpected for me, but now so now we could combine our work and their work, and then we had a good advance in our understanding of immune tolerance and also understanding of human immunological diseases. So just you continue, and then something happens. So you don't have to worry about whether you can find out something very big. Well, you don't have to. Just continue and then optimistic, and then you'll see that something happens. So always that is happens. That kind of things happens in science. So new technology, and then now you can solve your own question or new finding by others. So science of the time, that is not only your contribution but others' contributions. All together, it's progressing. So that is, I think, that is important.
So stick to it.
Stick to it, and to be optimistic.